This webinar is supported by the Brazilian Wheat Science Society, our foundations, CAPES, CNPq, and FAPERGS. And I would like to introduce to our webinar speaker, Dr. Nilda Burgos. She is from the University of Arkansas. We have been working together with several research experiments with Dr. Burgos since 2012, I guess, Dr. Nilda. And
yes, 2011.
11, yes. It's been a while that we work together. It's very good to see you, Nilda, and thank you very much for your willingness to give this speech to the students. And as I told you, we are broadcasting this to people all over the world. And thank you very much for that. And I will handle the floor to you.
Okay, Luis, thank you. Thank you so much for the invitation to participate in your seminars, in your lecture series. I am very glad that we actually have several people listening in. And although having this is really no substitute to the being there in your classroom and interacting in person with the students. But this is the best we can have, and we are actually thankful for having this technology that would allow us to still communicate over long distances and reaching a broader audience. So before everything else, I would like to say that I am not a chemist nor a biochemist. I am just a weed physiologist, basically trained in weed science. I had background in soil science, of course, as well. But this topic that we are talking about, I learned about this as I went through with my research pursuits and gathered information as I went along, and just put together the general information in a simpler manner. And I prepared this series of slides with Luis's students in mind, considering that we are coming from different backgrounds, and the students may not have, of course, the same level of experience, nor the same level of understanding. So I'm kind of putting this in much simpler terms. And I am not the only one who can actually talk about this topic. There are many other more qualified people who can talk about this topic. But we are just going to go through general principles, and hopefully this will provide the beginning students resources so that they can conduct research better and assimilate more knowledge, more information better, and screen or evaluate or assess quality of information that they are seeing in the literature as they move along in their learning process. Okay, so let me share my screen with you. And I don't know, let's see, how did we do that?
It's in the bottom of your screen. If you go in the bottom, you can see the share screen.
Like here. Where did we go? Share. Okay. So now, Luis, can you see this?
Yes.
All right. So let's start. first, let me introduce you very briefly to, well, it's not doing anything. How does it go from to the next screen? I think this is frozen, Luis. Let me.
It's not changing slides.
Yeah, trying to move to the next slide.
You may share your screen again and try again.
Okay, stop share. What is this? Okay, stop share and then share again. We just did this before. Okay. What is it? What is it doing? Okay, Luis, let me okay, now it is doing that. All right, so now we're able to move along. So the University of Arkansas is located here in the Mid-South of the US, and this is the state of Arkansas here for those who are tuning in from other locations where you're not familiar with where the university is located. So we are in the mid-southern US. And then our campus here is in Fayetteville, Arkansas, and we have an intermediate sized campus here, close to 30,000 students. But we have other campuses in the state. Now, the department where I am in is the Crop, Soil, and Environmental Sciences Department, and we have different areas of research roles here in the state in breeding, in physiology, and biotechnology and genetic engineering of crops. The biggest crop that we deal with here is rice, soybean, cotton, and wheat. And these are where we have strong breeding programs. We also have soil science in the department, water science, and environmental fate. But then where I am specifically is in weed science. So in weed science, we do research in all of different areas in this field. So biology, ecology, physiology, and all that. And I myself, I also do a little bit of molecular biology and weed genomics, in addition to just routine research on weed management and herbicide testing and all that. So into our topic on herbicide metabolism. I would like to start with saying that selectivity of herbicides in between crops and weeds, or its spectrum of activity, is really depending on different mechanisms by which plants respond to the herbicide or plant-herbicide interaction. So selectivity is achieved by different methods. In our weed management attempts, we try to achieve, for example, selectivity of some herbicides by altering placement of herbicide, separating it from the crop seed. And this is why some herbicides are recommended for application pre-emergence on top of the soil, and you place the seed below the soil deep enough, cover it so that it doesn't get directly in contact with the herbicide. In this case, selectivity is achieved by physical separation and not as much as due to physiological differences. So there are different ways by which plants can tolerate herbicide. And you can see this here listed, what is common we can encounter. So differential absorption, different rates of translocation, different sensitivity of the herbicide target, different rates of metabolism, and different capabilities of the plant to protect themselves from the phytotoxic action of the herbicide in cases where herbicides reach the target efficiently and are not metabolized fast enough. And then sometimes plants can survive that situation by eliminating or mitigating the phytotoxic action of the herbicide. Of course, all this, whether we achieve plant death, depends on the dose of the herbicide and how the plant can try to fight that. So from this context, we can actually now imagine that the ability of the plant to detoxify or metabolize the herbicide is one major way by which we can achieve selectivity and by which weeds can survive herbicide applications. Okay, so the reason for non-selectivity of some herbicides like glyphosate, paraquat, glufosinate is because their action is very difficult to overcome. And so it's either the plants cannot metabolize them fast enough, or the plants cannot metabolize them at all, or the effect of the herbicide is too much so that any protection that the plant has is not enough to protect them from herbicide injury. Okay, so in metabolic process, the protection from injury via metabolic degradation or herbicide metabolism is achieved by a series of processes. So in the plant, these are the kind of four categories by which plants are able to process herbicide molecules to make them non-toxic or less toxic. So it starts with herbicide breakdown or herbicide changes. That includes oxidation reactions, hydrolysis reactions, just starting from the very simple modifications that require less energy or easy to do, easy to achieve chemically or physically, like in less energy required for the plant to do it. So easy reactions, these are the first ones that will happen to the herbicide molecule. The next phase will be more difficult reactions, and this would be phases that would require enzyme activity, because then this will need help with facilitators. So enzymes are facilitators for herbicide breakdown. And then once the herbicide molecule is modified and broken down into different metabolites, then there are processes in the plant that would tag or conjugate or attach larger molecules to these metabolites, and not necessarily always large. Some conjugation reactions happen with amino acids, just these are which are smaller molecules. Okay, so then these conjugated molecules will be transported either to the vacuole for final digestion or degradation, or will be incorporated into cell walls. And so then that is how the metabolism in general works. And so what we already have learned so far is that differences in translocation of herbicide in the plant can be a factor or can be a manifestation of differences in metabolism, because when the herbicide is metabolized, then it would create a differential gradient right? So if the herbicide is metabolized, then it will not reach its target, and we would say that it's not translocated far enough, or it did not get distributed to the different parts of the plant, and we already know that that limited distribution in the plant could be a consequence of metabolism of the herbicide. Okay, so degradation of the herbicide in plants. So this is showing you the general steps that could happen, and the 4 processes are outlined here. Phase 1, conversions. Phase 2, conjugations. Phase 3, depositions into different places. And then phase 4 is the final processing of the herbicide metabolites. I would just like to point out that in some of these stages, the metabolites can exist in a form that is still in a reversible reaction, meaning that the modification is not totally unidirectional, that depending on the environment in the plant, that the metabolite can go back and forth into the original form versus the other form, and therefore would still remain to be in a toxic or phytotoxic phase in the plant, and will remain to be phytotoxic until the plant is able to process it further into a non-reversible form, and it will stay in a non-phytotoxic stage. So there are examples of that you will also see in the literature or when you study some specific herbicides. Here's some more description, like a descriptive way of looking at the different phases of metabolism. So phase 1 involves oxidations, basically, oxidation reactions, hydrolysis, and reductions of molecules. The biggest player in this phase 1 herbicide metabolism would be the cytochrome P450 enzymes. And in a short bit, we're going to talk about them quite a bit, because they are the major players in breaking down of xenobiotics in the plant. Hydrolysis reactions are carried out generally by carboxyl transferases or other enzymes that would facilitate adding of water molecule into the herbicide. So the general purpose of phase 1 reactions really is converting the herbicide molecule into more reactive forms, because in general, the herbicide molecule does not like water and they are less reactive. And then these oxidations, hydrolysis reactions are going to facilitate or increase the reactivity of the herbicide with water. And then phase 2 will be conjugations, and conjugations involve glutathione S transferases, because this basically conjugating the herbicide molecule with glutathione. And then there are also glycosyl transferases that are involved, because this would facilitate conjugation with sugars. And so these are big molecules that are attached to herbicide metabolites, making them permanently non-phytotoxic. And then there are secondary conjugations that will occur after that. And secondary conjugations could be further conjugation with other molecules. We would have examples, a few examples of this type here. And then this would also include further modifications by polymerization, meaning conjugation with another molecule of sugar, for example, make the metabolite even bigger. And then, of course, the last phase will be incorporation into the cell wall or further breakdown in the plant. So the cytochrome P450s, we're going to talk about this compounds more. So these are enzymes, so they're proteins. They're generally classified as mixed function oxidases. So they can perform oxidation and reduction functions, mixed function. All right, so these are, this is composed of many families. So in this group of enzymes, there are many families of enzymes here. Right now, there are more than 500 cytochrome P450 genes that are reported. And they belong to at least 50, actually now more than 50 known families of cytochrome P450s. And then what they do is represented in this reaction here, in this simplified reaction here. So the general reaction that they do, whatever it is, would be in this form. So you would consider the R group here, this R, you could imagine that to be any kind of compound. And then that has a hydrogen molecule here that is available, easily accessible for reaction. And then it reacts with oxygen. So it's oxygenation, right? So adds oxygen to that, oxygenates that compound. And then here would result in hydroxyl formation, attachment to that parent molecule, and release of water. And so this reaction here is energy consuming. It requires energy input. In this case, it's NADPH that is providing that energy. So it's a reaction that is not easy. It needs energy to push it forward. And this is what the P450s do. So here's an example of, so now we're going to go through some examples of herbicide conversion. So now the remaining series of slides would look at different examples of herbicide conversions in the plant. So here's an example of hydrolysis reaction. And this one here is specifically an aryl oxy-phenoxy propanoic acid herbicide. So this is under what we call AOPP herbicide family. This will be group 1. This would be under the ACCase inhibitor group. And this particular example is diclofop. There's diclofop methyl. So here we are going to review a little bit about terminology so that we all can follow. So here's aromatic rings are also called aryl rings, aromatic aryl. So here, this is where it is, it gets its name. Here's aryl oxy-phenoxy. And this side chain here actually has 3 carbons. So this one here is a propane, a propyl group. And so this one here is what is referred to as the propanoic acid group, meaning it's a propyl carboxylic acid. Here's carboxylic acid here. So anyway, this is an ester. So the parent molecule is an ester. And I don't know how to move this to the side, Luis, but anyway. An ester is an R group, which you can look at R molecule as this big molecule here. And then it will have a carbonyl group in the middle, which is this. And then an oxygen bridge, and then another R group here. So that is an ester. And so this hydrolysis here is hydrolysis of a carboxylic acid ester. And so this is hydrolysis of this particular particular part of the molecule here. This is the part of the molecule that is most easily accessible in this form. So when it is hydrolyzed, then you can see that now this side chain is shorter. The methyl group is gone. It's hydrolyzed to its carboxylic acid form. It's no longer a methyl ester. It is now a carboxylic acid. So in the plant, this is what happens. This is actually the one that is transported in the plant. This is the active form of the herbicide. The methyl ester is a formulation that would help it enter the plant. And then in the plant, it is changed into the acid form, which is the active form. So the plant has to do something about this acid form of diclofop. Now in the weed, here's wild oat. This is the weed. This is how it modifies it. In the weed, this acid is converted into an ester conjugate. But then this one here is reversible reaction. This is what we're talking about, a reversible phase of the modification reaction. So it's converted, wild oat converts this into this form, but so supposedly this form is non-phytotoxic, but unfortunately this reaction is reversible. So for as long as it can go back to the acid form, then it does not totally remove the phytotoxicity of the herbicide. So therefore, diclofop is going to kill wild oat. But in wheat, this diclofop acid is modified into this form where there is ring hydroxylation. So the modification of this aromatic ring and hydroxylation of it renders this ring subject to further modification, and then also renders this non-toxic to the wheat anymore because it doesn't go back to the acid form. So this is now non-toxic. There's further modifications here in order to make it, to further break it down. But as long as it is ring hydroxylated, it doesn't go back to the acid form, and then it is no longer toxic to the plant. So wheat is tolerant, but wild oat is susceptible. So it's kind of like how, in this case, this ring hydroxylation is facilitated by cytochrome P450. So another example, here's ring hydroxylation, but now with 2,4-D. So here, 2,4-D molecule is simple, so it is easier to follow. So here's 2,4-dichlorophenoxyacetic acid. So here's carbon number 1, so 1, 2, 3, 4, so that's 2,4-dichloro. This whole molecule here is a phenoxy because it is a phenyl ring with an oxygen on it. And this part up here is the carboxylic part. So it's phenoxy carboxylic acid. So when it undergoes ring hydroxylation, which is facilitated by P450, where it is hydroxylated is here on the 4th carbon. But what we are seeing here is the final process. The process of hydroxylation is actually a little bit more complicated because in this case, this chlorine has to be moved to the 5th carbon and then replaced with oxygen and hydroxyl group over here. And so this is a more complex reaction actually. And eventually, because of this hydroxylation here of the phenyl ring, this is where it opens up for conjugation with glucose molecule. So what we're going to end up here is what we would call an oxygen glucoside. So an O-glucoside is the sugar conjugate. So that's what happens with 2,4-D. Then when we look at another molecule, which is also, it happens to be also a group 4 herbicide. So we're talking about some phenoxy-oxinic molecule here, 2,4-D being an auxin-like herbicide, and dicamba here, another auxin-like herbicide. So dicamba is also a simple molecule, so it's also easy to follow this example. So here we're looking at 3,6-dichloro methoxybenzoic acid, so benzoic acid. So here's carbon number 1, 2, 3. So that's 3, 4, 5, 6. So this is 3,6-dichloro. Methoxy is coming from here because this here, this substituent here is a methyl oxy. So methoxy substituent here. And then this is the carboxylic acid functional group. Okay, so one thing that happens to this molecule is that it could be acted upon by an O-demethylase. So where would an O-demethylase work? It would work on removing the methyl group. So here's the methyl group over here represented by this bond. This is a methyl group here. So that demethylase will remove the methyl functional group and then will expose oxygen and to hydrogen bond. So oxygen will bond with hydrogen. So we will have a hydroxyl group here instead of a methoxy group. So that particular group can be open to further reactions. The other thing that can happen to this molecule is also hydroxylation of the aromatic ring. So in this case here, hydroxylation of carbon number 5. So now we have a 5-hydroxy-dicamba in this case. But then eventually we can have these two changes happen with the methoxy, with the methyl group removed and also with hydroxylation of the 5-carbon. And then we will end up with 2 hydroxyl groups in this ring molecule. And we will end up, of course, with a non-phytotoxic version of dicamba. A third example. So now we are looking at another, so besides hydroxylation, there's demethylation. So demethylation just means removal of a methyl group. So methyl group that is attached to either nitrogen from an amino group or attached to oxygen. And so in this example, we have chlorotoluene or chlorotoluene. This is a substituted urea. This is a photosynthesis inhibitor. Talking about photosynthesis inhibitor here. The base structure of substituted urea is actually a urea molecule. So here's urea molecule here. Urea basically is a fertilizer, of course, and we have an amino group attached to a carbonyl group attached to another amino group. So very balanced molecule. Now here is the base molecule of substituted ureas. So in this group of herbicide, their basic structure is urea, and then they would just differ in what substituent is there among these 4 hydrogens. So there are 4 hydrogens that can be potentially replaced with anything that is found herbicidal. In this case, chlorotoluene has this ring aromatic substituent here that has a chlorine in it. Okay, so one of the things that could happen to this molecule is that so here's the substituent, and then we have the methyl group, and then we have the amino group here, and then here's one methyl group that is removed from from the other side. So see how there are two methyl groups here? So in this particular metabolite, one of these methyl groups is removed. And so this is what the process is that we're talking about, N-demethylation. So when you look at other herbicide molecules that are attached to an amino group with a methyl attached to an amino group, that can be potentially removed by this process. So not only chlortoluene, but many other herbicide molecules that have this functional group right here. All right. Then here's an example of I don't know what happened there. Ah, okay. So we have a question. Let's see. What? Oh, okay. You have your.
Nilda, I just started a poll, and
okay.
so you just need to move that from your screen.
Let's see. I don't know how to move this.
We can take some questions if you have time, and then
sure.
and people can answer the poll.
Okay, sure.
And after that, I finish the poll, and then you go out from your screen. So everybody, if anybody can need to answer, have some question, it can ask on the question and answer. Question from John Mota. Phase 4 occurs under the sun. I think he means that does it need sunlight to occur the phase 4 of metabolism?
Oh, no, it's not dependent on sunlight. It can happen any time. Mm-hmm. Although the plant's ability to finish all this metabolic degradation, of course, will be helped by having enough inputs, enough intermediates to facilitate it. So everything that requires transport and glucose conjugates, etc., all of those will be helped by a good physiological stage of the plant. But yeah, phase 4 does not depend on sunlight.
The other question is from Thiago Gazola regarding resistant herbicides, resistant weeds to herbicide. For glyphosate, what's your opinion about metabolism via GOX and aldoketo reductase?
Oh, okay. So on that topic, resistance to glyphosate. See, plants, what we know is that plants do not do that hardly. Well, actually, no. So because there is technology developed to produce glyphosate-resistant plants with this capability to do the GOX reduction, etc. But that is borrowing genes from bacteria. Bacteria can do this to glyphosate, but plants cannot do it. The one process that plants can do would be to break down glyphosate into AMPA, but that even is rare and limited, very limited in the plant. So that's why very few plants are really tolerant to glyphosate.
Yep, I think those are the questions we had. Oh, there is another one here.
Okay.
Francis asks how many families of P450 enzymes are there in plants, and are the active sites of these highly conserved between different plant species?
So what we know is that there are more than 50 families of cytochrome P450. In the literature, they are numbered. So their numbering, well, their numbering is not all, their numbering is not very smooth or consecutive, but the nomenclature is improving. As we speak, because now with better tools, we are getting more and more understanding about cytochrome P450s. They're so diverse. And so, for example, one family, I have an example that I will show in a few slides later. And for example, the family of cytochrome P450-81, there are many members of this, and there's another cytochrome CYP72, and also many members. And within each family, there's so much diversity. And what we know is that their diversity of function is, what I can see is that their diversity of function is really facilitated by the diversity of the members of each family. And as far as site, they're proteins, and they're really kind of, so they're enzymes. So it's the substrate that would bind to them. And so because they function as, one of their main functions is detoxification of compounds that are either in the plant that they need to detoxify, or compounds that are external, so exogenous, compounds that we apply, such as herbicide or insecticide or fungicide. And so what we know is that some of them have specificity, but others have cross-reactivity, meaning that they can react with multiple molecules. And of course, the cross-reactivity to molecules will depend on the similarity of the molecular structure of these substrates. So there is this. Now, as far as conservation, we don't know enough about the details of all of these to be able to say in general if they're all highly conserved, or if they are all very, or if they have high mutation rates. I don't know that. But in terms of the ones that we're studying that are already implicated in resistance to herbicides, there are not much information yet actually on the sequence of this particular P450s, for example. We just say, oh, in this resistant population, there is overexpression of this particular P450, let's say CYP72A1, for example, hypothetical. And then, so for now we say, okay, that's the one that is involved, and then we overexpress this particular P450 in bacteria or yeast or plant or Arabidopsis, and then it confers resistance. So that's that. And the sequence is not really reported as to whether this particular P450 is different in sequence compared to the same P450 from a susceptible plant. So what I'm saying is that the information that is building up now relates to expression level rather than sequence level. So we don't know much about sequence yet. That's what I could say so far.
Okay. Juvan he asked, frequently antagonism of auxin like herbicide and ACCAs type of herbicide is observed. There is some evidence that this can be related to P450 enhanced activity. Could this have any impact on resistant development?
Could it have any effect on resistance evolution? Resistance evolution to either herbicide, right? Either the auxin or the
ACCAs. Yeah.
Okay, what I would say is that anything that would modify the toxicity level of a herbicide and would cause that plant to survive that application and reproduce, then that becomes a factor for selection for resistance. So if there is antagonism that occurs and it reduces the efficacy of the ACCAs component, because that's usually what is compromised when you mix oxinic herbicide or other herbicide with ACCAs inhibitor, then the activity of the ACCAs inhibitor is compromised. Then it would favor selection for eventual resistance to the ACCAs herbicide. It may make resistance to ACCAs faster, to the evolution of it faster. But this is kind of a general principle that actually applies to any combination of factors though. That if you're compromising the activity of one and allowing this particular plant to survive that, then that is one agent for selection for resistance.
We have several questions, but I think we can move on and then.
Okay. Okay. And you can email questions also later. Okay, so we've talked about this in the methylation and then now, so I guess there is this problem about this, Luis, that if we stop for a while, they get stuck, okay?
Yeah.
So here, let me see. All right, so here's hydroxylation. Well, what happens to imazethapyr? So now imazethapyr, it's an ALS inhibitor. It is an imidazolinone, and it's just kind of showing us what happens to this molecule because it's a more complex molecule. So the first thing that happens is hydroxylation of the first carbon in the 5-ethyl substituent. So kind of takes a little bit of review here. An ethyl is an alkyl group. So it's a straight chain carbon with 2 carbons in it. So here's ethyl right here. So in imazethapyr, where is the ethyl substituent in the pyridine ring? So, well, number one, what is a pyridine ring? So here is, this is a pyridine ring. So pyridine is an aromatic ring with 1 nitrogen replacing a carbon in the ring. So this pyridine. The ethyl has 2 carbons. So ethyl is a side chain that has 2 carbons. So this is the ethyl substituent. And so it's like, okay, why is it called carbon number 5? Because in the numbering of this, we start with this position. So this is 1, 2, 3, 4, 5. So this is carbon number 5 with the ethyl substituent on the pyridine ring. So what happens to this molecule in the plant when it is tolerant is that it hydroxylates the first carbon. So, okay, this is the carbon number 1 and this is carbon number 2. So this carbon number 1 is hydroxylated, meaning the hydrogen is replaced with hydroxyl group. And then with this hydroxylation going on here, then it actually opens up a nice place where glucose can be attached. So this is O is going to form an O-glucoside, because now glucose will attach to this oxygen here, will oxygen bond there, then it will produce an O-glucoside. But anyway, in the susceptible species, this thing will not happen. That's how they survive, emulsifapyr. Okay, other kinds of oxidation reactions. So basically oxidation, this is what I learned from my basic soil chem class. So oxidation. So oxidation involves loss of electron. So Leo, loss of electron is oxidation. So this, I learned this from my soil chemistry professor when I was in college. Oh, actually in grad school. So loss of electrons, whenever there's loss of electron, then it's oxidation reaction. But anyway, so one example is deamination of triazenones. So triazenones, so here's a triazine, 3 nitrogens in the ring. It's an one because it's got a ketone group here. So it's triazinone. This is metribuzin. So when it is deaminated, meaning an amino group is removed. So it's talking about this amino group right here. So then when you look at that here, this is now that nitrogen minus the amino group. Okay, so this is deaminated metribuzin. So then other things happen, and we have here the 2 nitrogens in the ring, and we have this nitrogen that used to have an amino group, and then we have now 2 oxygens in the ring. So this is now non-phytotoxic form of metribuzin. But the thing with this is when you have this oxygen, it's an oxygen or a nitrogen exposed, which is going to be more available for bonding, then that is where conjugation happens. So in this case, it's conjugation with sugar, with glucose. But that glucose, in this example, instead of conjugating, bonding with an oxygen, it formed a bond with the nitrogen. So now this is an N-glucoside that is being formed in this example. So this is now non-phytotoxic group form of metribuzin. So tomato, which is a very well-known vegetable that is tolerant to metribuzin, forms this particular metabolite, N-glucoside, and soybean as well. So that's that. Now, other kinds of conjugation. So now here's atrazine. Now this is atrazine. This is triazine, balanced distribution of nitrogens here, but this is chlorinated, chlorinated triazine. So this is atrazine. So one of the things that can happen to it, of course, is replacement of this chlorine molecule with an oxygen. So here's that. And then when it is oxygen here now becomes available for bonding with something else. And what is bonded to that in general is glutathione. So here's glutathione. Glutathione is actually a peptide. It is a peptide. It's a tripeptide. So it's peptide with 3 amino acids, glutamate, cysteine, and glycine. The useful component of glutathione is this one here in the middle. So cysteine has sulfur in it. And sulfur is very useful because sulfur can accommodate a wide variety of charges. So it can accommodate different range of electron numbers, and so it is ideal for bonding with a lot of things. And so then now it forms, so here's the sulfur bridge, and now it's forming a bond with this triazine via this exposed oxygen molecule there. So this is now the atrazine conjugate, glutathione conjugate Okay, so glutathione is used in this manner, but it's actually used it is actually used as an antioxidant in plants a lot. So glutathione is very useful for that protection from reactive oxygen species. And glutathione is being used actually in the cosmetic industry now by people who want to reverse aging. I don't know how effective that is. But that's kind of what glutathione can do. Now, conjugation with glucose, there's another example here, picloram, a very simple molecule. This is also a pyridine molecule. This is under the pyridine carboxylic acid group of herbicide family. And so here's the pyridine molecule conjugated with glucose here, okay? And then I'm showing this as an example of a polymerization reaction. So now here's the first glucose, and then it is polymerized by adding another glucose. So this now is a secondary glucose conjugation. So that's one example there, okay? So here's this one here is kind of just showing that we also have an example here of an N-glucoside, and we also have an example of an O-glucoside. So like different kinds of glucosides. So secondary conjugation can also happen with another molecule, not just glucose. So here's an example of a glucose molecule that was the N-glucoside from that picloram from the previous slide. And then that N-glucoside was underwent and can undergo secondary conjugation with malonate. So now this is the secondary conjugate here. So in review, there are two kinds of glucosides. We have N-glucosides and we have O-glucosides. And these glucosides can undergo secondary conjugations to further the detoxification of the herbicide molecule. Okay, so the metabolites, like we mentioned before, in phase 4, phase 3, phase 4 involves transport and then deposition and final degradation. So they can be deposited in the vacuole, which is in this example, here's a mature cell, very large central vacuole. There are other vacuoles, but normally there's vacuole is the digestive organ of the cell. So a lot of compounds that are here, there are many acids here that can degrade all sorts of molecules. And then also some of the conjugates can be deposited into the cell wall and be incorporated there as cell wall materials, or they can be degraded into their amino acid forms and all that and used in other processes. All right. So, well, before we go in here, do you have other questions? How much time do we have?
We have time. We have a question from Cássio Mendes. Okay. When the herbicide is metabolized, can it be bound residue in the plant similar to what happens in the soil?
Can it bind some other molecule in the plant?
Yeah, like in cell wall, or can be a bound residue?
Yeah, it can be a bound residue so where it can be bound would be, yeah, it wouldn't be in structural components like we said here. It can be excreted into the cell wall and be bound in the cell wall materials. Structural, I guess, is what my understanding of this is. It can go into structural places and just be held there. Because if it goes into the vacuole, then it gets digested into its basic components, into amino acid or elemental components, something like that.
We have another question from Caio Brunharo. How specific are these enzymes? Do they target a specific compound or class of compound? For example, if a P450 metabolizes chlortoluron, will this same P450 enzyme also metabolize other
Substitute urea? Oh, right. So, so yeah, that is a common, that is actually a logical thought process that we say, okay, if this particular P450 metabolizes chlortoluene, then can it metabolize something like oh my word, my mind just went blank of other substituted ureas. So our normal deduction would be that if the structure is similar enough that it would be cross-reactive to that the enzyme will also bind that. There are actually examples of, or several examples of P450s that metabolize different herbicides just because of structural similarity. And in the literature, there are already some list of P450s that can metabolize different ALS inhibitors, for example, such as sulfonylureas. Can be, then we can say, okay, this P450 metabolizes this range of sulfonylureas. We will see an example. I actually put here one example of this P450 family and its cross-reactivity to different herbicides, and that would be a good one to show to demonstrate this. So they have, some are, well, what we're seeing in different research is that some are more specific and others are more cross-reactive. And what I know so far is that it is difficult to, what is that? It is difficult to generalize with P450s. There are just a lot of diversity.
Mm-hmm. Another question, maybe we'll take just another one and then we can move forward. Mm-hmm. Dr. Martin via Ayub. Hi Nilda, the lack of toxicity of metabolites when they are conjugated with glucose, is it because they increase in molecular size or cannot bind into the target enzyme?
Yeah, that's basically the principle, is that well, when they're attached to a big molecule, then of course they won't fit in the binding site anymore. And then conversely, if they're broken into smaller molecules or made more polar or vice versa, their configuration is different, then they could be loose, meaning they could enter the site, but they're not held strongly there anymore. And a lot of the natural substrate interaction with herbicide involves competition in this binding pocket. So whatever modification happens that would alter the bonding configuration of the herbicide molecule in this pocket will cause the herbicide not to be active anymore, or less, we say, okay, less phytotoxic or totally non-phytotoxic, because of this. It either loosens the binding in there, making the natural substrate the stronger fit, or it's just big that it couldn't even enter the binding pocket anymore.
That's it.
Okay. So I just want to touch on this very briefly because herbicide safeners act by either enhancing detoxification reactions, basically, and where a lot of examples really would be enhancing the expression or production of glutathione, for example, or GSTs. So these molecules that are conjugates or protectants, that help the plant. So yeah, so here I've listed some of the items that can happen if these safeners are used, whether they're used with herbicide application if it is foliar, or whether they're used as seed treatment. So it actually has something to do with increasing the metabolic degradation of the herbicide, or increasing protection from herbicide phytotoxicity. So what we know so far is that, so some of these examples here, binoxacor for metolachlor in corn, cyumetronil for metolachlor in sorghum. What we're seeing so far in terms of commercial applications of this would be in the monocot crops. For some reason, they are more effective in the monocot crops in certain herbicides. All right, so just kind of throwing in here some examples of these safeners, and sometimes it is kind of easy to see that some of these safeners work with certain herbicides because of structural similarity, but others are not really just because of structure. But these are kind of just some examples of the popular safeners that are used to facilitate herbicide degradation in the plant, so that you can achieve selectivity for crops. You have to be careful in doing this, and so it requires, of course, a lot of research to achieve a good balance between increasing degradation of the herbicide, safening the crop, versus still obtaining the level of weed control that we need. So there's a balancing kind of thing there. So there's this paper by Dr. Rickers about explaining the activity of how safeners actually act so that it can defend the plant and help detoxify herbicides. It's kind of just here looking at the safener here, and then the different places where it can go, and then how it can modify. Because of these factors on top, it can modify the expression of these protection genes, so GSTs, and it can even increase, P450 expression, and then it can affect higher herbicide metabolism and impart tolerance, greater tolerance to the herbicide. And so we desire this for crops and not for weeds, and so there's that. So I would just like to touch a little bit on this because with respect to herbicide metabolism, this is the normal consequence. So this is the logical, ecological consequence. When we're selecting plants or weeds that are able to survive herbicide application via metabolic degradation, then we end up with this non-target site resistance. So the thing to remember is that whatever it is that allowed plants in the field, just natural population, to survive a herbicide application or a low-level herbicide application, those same mechanisms are the same mechanisms that we are selecting if we are subjecting a large enough size of population of weeds to this selection pressure, or if we are, if they receive sublethal doses by unintentional reasons, unintentional ways, then we are selecting for these same mechanisms to achieve tolerance. We accumulate these genes, protection genes, low effect genes, but then we accumulate them with time, and then we end up with non-target site resistance. And this is difficult. So metabolic NTSR. So we end up with non-target site resistance by metabolic degradation. So how can this arise? So it can happen one of three ways. Maybe there are more, but I've listed here three. So one way it can happen is, we have mentioned earlier, are these P450s highly conserved, meaning are their sequence, are their gene sequence, nucleic acid sequence highly conserved? Well, it is possible that that some of them are not highly conserved. And even if they are highly conserved, it is possible that with intense selection pressure, we are able to select the rare mutants out there that would be the resistant mutants, okay, or the better P450s, right? So that's possible. Another way is that we can increase the activity. We have selected more efficient version of the same P450. So how can it be more efficient? Well, we don't know. Greater efficiency would mean would actually relate to a favorable mutation in terms of the enzyme function. So it could be a mutation also that happened over time, accumulated over time, and then or just exists in nature, rare event, that there is a version of this particular enzyme that is an isozyme that is more efficient, has higher Kmax, whatever the enzyme kinetics is. So that's possible. We could select that, or we could also select for increased production of the P450. In this case, it is not the P450 that changed itself, but something that controls its coding or controls its production. So it could be something that happens upstream, could be a transcription factor change that affects higher production of the protein in the resistant population, right? So, or when it is challenged with the herbicide, then it is able to produce more of that particular P450 compared to the susceptible population. So far, what we are seeing is number three. I mean, what we kind of see is that when it is challenged with the herbicide, then it is producing more of this P450 compared to the susceptible population. Or without any herbicide challenge, particular resistant plant already has higher level of this particular P450, what we call constitutive overexpression of a particular enzyme. So number three is what we are seeing so far. And the others probably exist. For example, increased efficiency, probably there, but of course that requires a different kind of experimentation, right? It requires biochemical experimentation, analyze enzyme efficiency. Okay, where are we seeing herbicide resistant weeds with metabolic resistance? Well, almost everywhere in the world now. It's increasing in cases. So in the Echinocloa genus in itself, there's a lot with Philopogon, Crusgali, Clona, to a lot of herbicide modes of action, a lot of NTSR, metabolic NTSR there. We know we have this in Rigidum, in Meiosuroides or blackgrass, and then of course in the Amaranthus genus with Palmeri and Tuberculatus, metabolic resistance to some herbicides. So we are seeing this more and more. That is the problem. Here is what I'm referring to earlier as an example of, this is work which is very good work by Dr. Iwakami in Japan and his PhD student. And this work actually, this paper actually won the first prize for paper competition in the Asian Pacific with Science Society meeting last year. And so this is studying the 81 family of P450, so CYP81. And can you imagine like how many herbicides they studied here, they tested here? So what they did was expressed various members of this family. Look here, they only look at 1, 2, 3. Well, I say only, but this is a lot of work, okay? Can you imagine multiplying all of this with the number of herbicides? So there's a lot. So there's 1, 2, 3, 3, 6, 9 kind of members. So they tested these different P450 genes in this family, okay? So the red ones indicate, high resistance. So up here would be the fold genes, and we can say here, the higher that fold is, the higher the resistance factor is. And so when it's red, then it is highly resistant. So for as long as it is yellow, so here, for as long as it starts being yellow, the fold gene is 2.5, so that means that it is getting to be on the resistance side here. So you can see each gene, its cross-reactivity. So each column here, if you look at this column, for example, this first column right here, talking about one gene in this family. So you see how many herbicides it is kind of cross-reacting. It is reacting with this. This is sulfuron. It's most reactive with that, but then it's also reacting with other ALS inhibitors. So this is ALS right here, right? So, and then it is cross-reacting some with here. These cross reactions are weak, but it is there enough that it makes it possible for selecting for resistance for these, for stronger resistance for these. So if you expose this population if you expose a weed population to these herbicides, they can be selected for resistance to these where there is already some cross-reactivity, right? And then so you can go down that. Here's another family here, another gene, A21. That is showing the same cross-reactivity as the first one. It's the first gene. Then here's this, second to the last, that has kind of weaker cross-reactivity, but also still cross-reactive to a lot. So you see, and then there's some that you can say, okay, there may be some here that is not really factoring very much. So this one here is not performing anything, so it's not a threat. This one here also is less of a threat, but there are 3 here that are kind of more important, and then here's a 4th one right here. So one thing is there's a lot of cross-reactivity in this group, but then you can also see others here, okay? So now the lesson is that with P450s, and considering that there are many families of P450s in a plant, not just this one, there are so many possibilities. So the potential for selection for metabolic resistance and cross-resistance to the same mode of action or multiple resistance to other modes of action, that is very high with this involvement of this, okay? So anyway, that's kind of like my closure slide there, just to look at metabolic resistance in general, and just even looking at cytochrome P450s and the complexity that this particular phase 1 enzyme group is introducing into resistance evolution and weed management. So what I would like for you all to remember, I wish that you would kind of just take this home with you as a general principle, is that the selectivity of herbicides is really governed by differential tolerance across species. So species have differential tolerance to herbicides, and this tolerance level is modified by a lot of factors: growth stage, environment, soil, temperature, etc., light, and all that. So that's modified by that. And the same mechanisms that endow tolerance to xenobiotics or herbicides are exactly the same mechanisms that we are selecting for resistance. When we apply the same herbicides often enough, broadly enough. And the problem with metabolic resistance is that the cross resistance and multiple resistance possibility is impossible to predict. It is so unpredictable. And because of this, it is also already happening that some of the herbicides that are newly introduced into the environment, into the weed population, newly registered, that there are weed populations now that already have multiple resistance because of metabolic and other mechanisms. That these populations are actually going to be already resistant to the, or tolerant, they become tolerant to the new herbicide that is introduced in the field. So this is our challenge. This is a major challenge for us moving forward. I have included references here to start you with, but there are many, many others there, but you can explore, you can start exploring these, and then I have cited some of those in the slides, but then there are many other resources available now. So if we have time, Luis, you can now control the time by ask questions or address discussion points.
Yeah, we can answer maybe two or three questions, and then we can adjourn. Thiago Gazola, he asked, under the field condition, is it possible to reverse the resistance of weeds that have metabolism as a mechanism? Example, malathion and mesotrione and on metabolism via 450?
Yeah, so we have a few good examples of this technology of, well, they kind of fall into a general category of using a synergist or something. And so actually there is one example is in Echinocloa resistant to propanil. And one of our weed scientists here, and Dr. Jason Norsworthy, when he was a student, he actually did his, one of his research studies was on looking for possible synergists for propanil that would help increase its activity on resistant Echinocloa. And so actually, and then other people also in other, maybe some scientists in Central America and Asia also explored this. So some synergists would be, yes, malathion is one, but in the case of propanil, it was an insecticide that worked, well, malathion is an insecticide too, but it was another insecticide, piperonil, something like that. And so then they came up with a new formulation of propanil that has some amount of insecticide in it that increases its activity. Now, that coming up with that formulation required a lot of experimentation because you have to balance increasing the activity of propanil versus not causing a lot of injury on the rice, because rice can metabolize propanil very quickly, naturally. Echinocloa cannot. The resistant Echinocloa is resistant because it had acquired ability to metabolize it. So now that's one example. So yes, it can be done. Just need to find the right combination of P450 inhibitor and the herbicide.
We're gonna answer the last question. Leonid asked, could you please explain a little bit more about the translocation versus metabolism of herbicides in different species?
Translocation, and okay, so I'll just I'll just address relationship between translocation and metabolism. So if a herbicide is applied in a plant that is able to metabolize it fast enough, then as the herbicide is deposited, so if we're talking about foliar herbicide, okay, so foliar herbicide is applied into the leaves and then it's absorbed in the leaf. If it is metabolized fast enough, it cannot really move far into the other tissues of the plant because it is already broken down. It will move, but in a different form. It's already broken down there close to in the cells where it got deposited. So that's why sometimes if we don't link the two, we will conclude that the resistance is because of reduced translocation. And probably what we don't know is the reduced translocation is because the plant is metabolizing the herbicide fast enough. So then it cannot go further to the other parts of the plant. Now, if it is reduced translocation per se, then we will see the herbicide in its intact form in different places, and we will probably see that that is happening because the plant is not metabolizing it. Does it make sense?
Yeah. Nilda, I think we need to adjourn. Thank you very much for your time and for your talk. And I will end up the webinar. Thank you very much, and hope to see you soon.
Thank you, Luis, and thank you all for participating, for listening, and I appreciate your questions. Have a great day.
Thank you. Bye-bye.
Bye.